Hello students, in the previous lecture I completed fermentation, that is anaerobic respiration, and we also studied about the output or balance sheet of anaerobic respiration. Now let's start with aerobic respiration. So aerobic respiration occurs by three steps. They are glycolysis, Krebs cycle and ETS. So glycolysis is the common pathway of both aerobic and anaerobic respiration, and we studied that glycolysis in the previous class. So by the end of glycolysis, one molecule of glucose undergoes partial oxidation into two molecules of pyruvic acid, which is released into cytoplasm of the cell. Now from the cytoplasm of the cell, these two molecules of pyruvic acid enters into mitochondrial matrix, where they undergo oxidative decarboxylation. Now let's study that step, that is oxidative decarboxylation. So during oxidative decarboxylation, two molecules of pyruvic acid, that is 3-carbon compound, undergoes oxidative decarboxylation into two molecules of acetyl coenzyme A. And this step is catalyzed by the enzyme pyruvate dehydrogenase. So this pyruvate dehydrogenase is a multi-enzyme complex. This PDH, that is pyruvate dehydrogenase, is a multi-enzyme complex. Multi-enzyme complex because it consists of three enzymes in it. Those three enzymes present in this PDH are pyruvate decarboxylase or pyruvic acid decarboxylase, then dihydrolipoyl dehydrogenase, and then third enzyme is dihydrolipoyl transacetylase. Now let's see the functions of all these three enzymes present in the multi-enzyme complex, that is pyruvate dehydrogenase. Okay, so this is the structure of pyruvic acid, which is 3-carbon compound. Okay, now this pyruvic acid undergoes decarboxylation, that is by the activity of pyruvate dehydrogenase. So due to decarboxylation, two molecules of carbon dioxide is released from two molecules of pyruvic acid. So two molecules of carbon dioxide is released. Where from this carbon dioxide is released? So this COO is released from pyruvic acid, which results in the formation of acetyl group. So this is acetyl group. COO is released. So CH3C double bond O. This is acetyl group. Acetyl group is formed by the activity of the enzyme pyruvate decarboxylase. Now, second enzyme, that is dihydrolipoyl dehydrogenase. This dihydrolipoyl dehydrogenase present in the multi-enzyme complex, that is pyruvate dehydrogenase, is involved in oxidation or dehydrogenation of this pyruvic acid. So when pyruvic acid undergoes oxidation or dehydration, two protons and electrons are released. Now those two protons and electrons are transferred onto NAD+, so that NAD+ is reduced into two molecules of NADH plus H+. So that is the function of second enzyme, which is involved in oxidation or dehydrogenation of this pyruvic acid. Next, the function of third enzyme is What is that third enzyme? Dihydrolipoyl transacetylase. Its function is to transfer coenzyme A, the group coenzyme A, onto acetyl group, so which has become acetyl coenzyme A. So this part is coenzyme A. So this part is coenzyme A. This coenzyme A is with sulfur group. So now this coenzyme A is attached with acetyl group by the formation of thioester bond. Actually, what is ester bond? If the bond is present between this one, so COO, this bond is called as ester bond. This bond is called as ester bond. Whereas the bond which is formed between, like this, C double bond O and S, which is the part of coenzyme A, this is called thioester bond. So what is happening? This enzyme, that is trans-acetylase, is involved in transfer of this group coenzyme A onto acetyl group, which results in the formation of acetyl coenzyme A by the formation of a linkage called thioester linkage or bond. So in this way, 2 molecules of pyruvic acid is converted into 2 molecules of acetyl coenzyme A by the activity of an enzyme pyruvate dehydrogenase, which is a multi-enzyme complex. And for the activation of this multi-enzyme complex, that is pyruvate dehydrogenase, 5 factors are required. 5 factors. 5 factors are required. These 5 factors are NAD+, Mg+2, TPP, thymine pyrophosphate. NAD+ is nicotinamide adenine dinucleotide, magnesium ions. TPP is the thymine pyrophosphate, lipoic acid, and coenzyme A. So these are the 5 factors which are required for the activation of this multi-enzyme complex, that is pyruvate dehydrogenase. Okay, this pyruvate dehydrogenase is first isolated from the prokaryotic cell, that is E. coli, and its size is several times more than the size of ribosomes. So this is all about oxidative decarboxylation, during which 2 molecules of pyruvic acid, that is 3 carbon compound, undergoes oxidation into 2 molecules of oxidative decarboxylation into 2 molecules of acetyl coenzyme A, which is 2 carbon compound, which enters into cyclic pathway of Krebs cycle, during which it undergoes complete oxidation into carbon dioxide, water and energy, and with the release of energy. So before studying that Krebs cycle, let's study the output and the balance sheet of this oxidative decarboxylation with respect to the formation of ATP, formation of water molecules and formation of carbon dioxide. So this is all about oxidative decarboxylation. Hope you understood this concept. Thank you, students.